Good morning, dear students. Our video today is the last one in the nervous communication. It's the second part of the synapses. At the end of this video, you will be able to identify the two functional types of synapses and compare between the effects of a microinjection of acetylcholine and that of Curer at the level of a synapse. Let's first remember the structural types of synapses. We have two types. If the synapse is between two neurons, like this one, it's a neuroneuronal synapse, while if it is between a neuron and a muscle, it's a neuromuscular synapse. But do all synapses have the same function and do all of them provoke the transmission of a nervous message? Let's see. So we have to study the functional types of synapses. So we make two experiments. First, we make the same excitation on both nerve cells. I have first nerve cell and second nerve cell in both experiments. Between them, I have this neuroneuronal synapse because it's between two neurons. Then we record the obtained results. In the first case, we obtain this recording. This recording is called depolarization, while in the second case we obtain this recording which is called hyperpolarization. So when we obtain a depolarization, the synapse here is called excitatory synapse, while when we obtain this recording or the hyperpolarization, the synapse in this case is called inhibitory synapse. So, as a summary, I have two functional types of synapse. The first type is called excitatory synapse because it allows the transmission of the nervous message by causing a depolarization, like in the first case, while the second type is called inhibitory synapse because it prevents the transmission of the nervous message by causing hyperpolarization, like the second case.
The neurotransmitters bind to specific ligand-gated ion channels in the postsynaptic membrane. A graded depolarization or hyperpolarization will occur, depending upon which channels open to ionic flow. Depolarizations, the result of sodium gates opening, stimulate the generation of action potentials. These depolarizations are known as excitatory postsynaptic membrane potentials, or EPSPs. Hyperpolarizations, the result of chlorine or potassium gates opening, inhibit the generation of action potentials. These hyperpolarizations are known as inhibitory postsynaptic membrane potentials, or IPSPs.
The last part of our video is to interpret some experiments. So, pause the video, open your book on page 77, read the experiments of document E, and let's interpret them together. Before starting the interpretation, what is acetylcholine? It is an organic chemical that functions in body of many types of animals, like us humans, as a neurotransmitter to send signals to other cells, such as neurons, muscle cells, and gland cells. First experiment of document E. The microinjection of acetylcholine at the level of synapses. So we have to inject acetylcholine here at the level of a synapse. It's a neuromuscular synapse since it's between a neuron and a muscular cell. Provokes a muscular contraction that is proportional to the concentration of acetylcholine even without exciting the nerve fiber. So only when we inject acetylcholine here, the muscles will contract without exciting the nerve fiber. This indicates what? This indicates that acetylcholine allows the transmission of the message to the postsynaptic membrane and causes muscular contraction, and this contraction is modulated by the concentration of acetylcholine, which means that when the concentration of acetylcholine increases, the muscular contraction will increase. The second experiment, the injection of labeled acetylcholine shows the presence of radioactive acetylcholine on the postsynaptic membrane. So, when we inject acetylcholine here, which is labeled, we see the radioactivity here on the postsynaptic membrane, which means that the acetylcholine binds to the postsynaptic membrane receptors. Now, what is Curer? It is a drug belonging to a family of organic compounds. It is used in modern medicines as skeletal muscle relaxants for general anesthesia and certain types of surgery. Now we have to interpret the third experiment. The microinjection of curer, not of acetylcholine like the first and the second one, at the level of synapses, prevent muscular contractions. So when we inject curer instead of acetylcholine, the muscles will not contract even if the nerve was effectively excited. So this indicates that curer prevents the transmission of the nervous message from the nerve fiber to the muscle. In the last experiment, when we compare the shape of acetylcholine and that of curer, we notice that there is a resemblance between the 3D structure of curer and that of acetylcholine in part of their molecules. So this indicates that curer can bind to the receptors of acetylcholine and block the transmission of the nervous message. As an evaluation, you have to choose the correct answer.